Good morning, guys. Good morning. Welcome. So if you could put your name in the chat as your attendance, that would be great. We are going to get started with our first lesson in Module 6, which is our special senses. So we finished up the nervous system. We talked about how neurons fire, how our brain is structured. We talked about our peripheral nervous system with our cranial nerves and our spinal nerves. And now we're going to talk about our special senses. And so we'll start, and let me share my screen here. So if you want to open up your Class 11 agenda, Class 11 agenda. So if you are on the Canvas page, if you see you're in Module 6, Special Senses, if you want to open up the Class 11 agenda and take the Class 11 attendance quiz. And if you guys could give me a definition of what is a special sense. What is a special sense? This whole unit is about special senses. And what do we mean when we say a special sense? So you guys take a minute and answer that. Double check who is here. What do we mean when we say a special sense? Okay, so our special senses are the senses that have a specialized organ for them. So for example, vision, which is the sense we're going to be talking about today, has our eyes as the organ that is responsible for vision. Sound has our ears, smell has our nose, and taste has our tongue. We also have a sense, two senses that are localized to our ears. So not just hearing, but also the sense of balance or equilibrium. And we'll talk more about those next week. The major sense that isn't considered a specialized sense, it's called a general sense, is the sense of touch. And that's because touch is localized all over your body. Okay, so we're going to be talking about our special senses. And we'll start today by talking about vision. And you can think of your eye kind of like a camera. So if anyone has used one of those like big fancy cameras, not the camera on your phone, for example, but one of the nice ones that has the attachable lens and you wear around your neck and you show everyone your big fancy camera. So we want to think about that camera when we think about our eye. So with a big fancy camera, we have a lens in front that we're going to adjust, and that lens helps the camera focus. Now on your phone, you just kind of like tap it to focus, but focusing is still done in the lens in the camera of your phone. The same is true with our eyes. Our eyes have a lens, and the lens helps focus the light onto the back of our eye, where we have a structure called the retina. So in a camera, the lens focuses the light onto either the film or the digital computer chip reader thingy. I'm not great with computers, but I assume there's some sort of digital thing in there. But with our eye, we're going to focus that light onto our retina. And our retina has two different types of photoreceptors, which are cells that respond to light. And these are neurons, and so they're going to fire an action potential when they receive the appropriate light signal. So our photoreceptors are neurons in our retina that will fire an action potential to respond to an appropriate light signal. And we have two kinds of photoreceptors, and we're going to talk about mainly cones today, but we also have rods. And rods help us see in black and white and in dim vision, and they're located more on the peripheral of your vision. And we are going to remember cones because cones are able to see, and they help us see color vision, and they're localized in the center of our eye. So cones with C for color, C for center. And so what we're going to do today is we're going to watch 4 short videos about vision and color vision in particular. And we're trying to understand the overarching question, how do we see color? Okay, so we'll spend some time going through that. And then for the rest of the class, we'll work on part 1 of a case study. Okay, so if there are any questions, please unmute yourself and ask or put them in the chat. And otherwise, I'm going to jump to the chat open. I'm going to jump to our first video here. Pull this over here. You know, there's just like a lot. I always have a lot of tabs. It's probably not the best way to live your life. Okay, this is the first one. So this is an overview on how we see color. Okay, let me unmute it here. So please, while you're watching these videos, take notes because again, you're gonna be submitting your notes from the videos and your case study. So please pay attention and take notes.
You might have heard that light is a kind of wave, and that the color of an object is related to the frequency of light waves it reflects. High-frequency light waves look violet, low-frequency light waves look red, and in-between frequencies look yellow, green, orange, and so on.
You don't have to get worried about the physics of light. So don't stress if you don't fully understand the wavelength light thing. This is mainly to show you when we get into the biology. So I'm more concerned that you understand the biology of vision than the physics of light.
You might call this idea physical color, because it says that color is a physical property of light itself. It's not dependent on human perception. And while this isn't wrong, it isn't quite the whole story either. For instance, you might have seen this picture before. As you can see, the region where the red and green lights overlap is yellow. When you think about it, this is pretty weird. Because light is a wave, two different frequencies shouldn't interact with each other at all. They should just coexist, like singers singing in harmony. So in this yellow-looking region, two different kinds of light waves are present. One with a red frequency and one with a green frequency. There's no yellow light present at all. So how come this region, where the red and green lights mix, looks yellow to us? To understand this, you have to understand a little bit about biology, in particular about how humans see color. Light perception happens in a paper-thin layer of cells called the retina that covers the back of your eyeball. In the retina, there are two different types of light-detecting cells, rods and cones. The rods are used for seeing in low light conditions, and there's only one kind of those. The cones, however, are a different story. There are three kinds of cone cells that roughly correspond to the colors red, green, and blue. When you see a color, each cone sends its own distinct signal to your brain. For example, suppose that yellow light, that is real yellow light with a yellow frequency, is shining on your eye. You don't have a cone specifically for detecting yellow, but yellow is kind of close to green and also kind of close to red, so both the red and green cones get activated and each sends a signal to your brain saying so. Of course, there's another way to activate the red cones and the green cones simultaneously, if both red light and green light are present at the same time. The point is, your brain receives the same signal regardless of whether you see light that has the yellow frequency or light that is a mixture of the green and red frequencies. That's why, for light, red plus green equals yellow.
Okay, so I'm going to stop that one there. So what I want you guys to understand from this video is that we see color vision from our cones. And we have three kinds of cones. We've got red cones, green cones, and blue cones. And when we're seeing different hues of color, it's the addition, it's both types of cones firing simultaneously. So when we learned colors back in like preschool or kindergarten or whatever, you learn that your primary colors were red, blue, and yellow. But for your eye, the primary colors are red, blue, and green. And to get yellow, it's red plus green. Okay, so again, in your retina, you have these cones. They're going to fire when they see the specific color that they're programmed for, and they're going to send that information to our brain. So we're now going to, do we have to take notes to turn in? Yes, you do have to take notes. So yes, you should be taking notes on these videos. Let's now talk about color blindness. So what happens when this process doesn't work?
Hey, and welcome to Brain Stuff. I'm Josh Clark, and this is the Brain Stuff where I explain to you how color blindness works. Have you ever been looking at a rainbow and thought, man, what is it like to be colorblind? That's a really weird thing to think, but here's your answer. First, though, we should say it's actually not called color blindness. The preferred term is color deficiency. So let's get a little more PC, shall we? Now, before we understand how color deficiency works, we have to understand how regular color vision works. And in humans, it's called trichromatic vision because we rely on three different types of cones, which are specialized cells in the eyes that contain photopigments. And photopigment is a protein that's sensitive to a certain wavelength of light, aka a color, right? So these three types of cones are attuned to short wavelengths, which picks up blues, medium wavelengths, which picks up greens, and long wavelengths, which is in the red band of the visible light spectrum, right? So when everything's operating correctly, our eyes are exposed to light, and the different cones send information to the brain to let it know what kind of quality and amount of the type of light that it's sensitive to it's receiving right then. The brain takes all this information, puts it together, and all of a sudden, these 3 different color informations equal 1,000,000 different shades of colors that we're capable of seeing. But what happens when it goes wrong? And you have color deficiency, not color blindness. What you have is what's called anomalous trichromacy, which somebody said is the most beautiful combination of words in the English language. I think it was that. In most cases, color deficiency is the result of cones just not operating quite right. And most of the time, it's either the red cone or the green cone. And the problem is that either the red cone or the green cone will mimic the other one. Rather than enhancing the cone that it mimics, it actually cancels out or mutes the information from the other cone. So the sum total effect of all this is that the good information coming from, say, the green cone is being canceled out or muted by the mimicky information coming from the red cone. And since the brain uses these different shades to make more colors, a lot of different stuff seems kind of dull or gray. That's color deficiency. Well, in particular, that's anomalous trichromacy, the most common type of color deficiency. The reason for all of this is genetic mutations, actually. Both of the genes that lead to the expression of cones in our eyes are found on the X chromosome, which is contributed by the female, aka your mom. Now, if your mom has a mutation on one of her genes, you have a 50% chance of some sort of color deficiency. If she has a deficiency on both genes, you have a 100% chance of having a color deficiency, which is what's called certainty. And what's even stranger is that a lot of people who are red-green color deficient don't know that they are until they're tested for it. Luckily, there are ways of curing this. There are contacts and glasses that can correct a little bit of the color deficiency, so you don't have to walk around feeling so glum and that everything's just kind of gray.
Okay, so color deficiency or color blindness, again, is caused by damage or a mutation to the gene for a cone, either your red cone or your green cone. And this mutation or this gene is carried on our X chromosome. So it's more common to show up in males because chromosomal males only have one X chromosome. So if they have a bad chromosome, a bad X, that's it, they're going to have this color deficiency. However, for females, chromosomal females, they have 2 X's, and so if they have one bad X, usually the other one can work and kind of cancel out the bad one, okay? So that's why color blindness or color deficiency is more common in chromosomal males than in chromosomal females. And the last thing we're going to do is we're going to do a little color blindness test to see if anyone is colorblind. My husband is, so he always finds these really annoying, but we'll just do a short one to see. So you should be able to see some numbers here. We'll do one more. Okay, and how these work, again, is because people who have the most common kind of color blindness can't tell the difference always between red and green. And so they would just see a bunch of circles, but we can tell, or people who are not color deficient can see the red standing out from the green. Okay, there is one last video we could watch. I'm not going to watch it this time. It's an interview. It's on the agenda. It's an interview with people who are colorblind, just about what their daily life is like. So we're not going to watch that last video, but you can if you are interested. Okay, so what we'll do for the remainder of the class is we're going to do this case study about vision and about a trip to the eye doctor. So I wear, as you could tell, corrective lenses, and at this point, glasses and contacts are pretty common. And so most people are going to the eye doctor for those reasons every year. If you don't wear corrective lenses, it's still recommended that you go to the eye doctor to help maintain the health of your eye. And sometimes we can have issues with vision, and it might take a little while for us to even know that that's what's happening. So what I'm going to ask you to do is I'm going to break you into groups, and I want each person to type up their own responses. You can talk in your group, in your breakout room, but you're responsible for submitting your own answers to part 1 of this case study. So there's 3 questions, and then you read a little more, and then there's another questions through 8, and then you read a little bit more, and then there are the last 3 questions. So we're going to go all the way up to question 11, and we're thinking about our eyes and how vision works. So again, our eyes have certain key parts to them. So you want to think about, as you, especially when you look at the structure and you're labeling the structure of the eye, don't just label it, but think, what is each part doing? As light is approaching the eye, what is the cornea doing? What is the pupil doing? What is the lens doing? What are all of these sections of the eye doing, and how do they contribute to vision? Okay, so I'm going to break you into breakout rooms. Again, you are responsible for submitting your notes on the video and your answers to part 1 of the case study. But I'm going to break you into groups, that way you can chat with each other about the case study and questions that you have. And then we'll come back together at the end and discuss the answers. Okay, so let me break you into groups. So those are open, so you guys jump into your breakout room and work on your case study. Again, part 1 through question 11. Do you guys have any questions? Is that why you're sticking around? Patience? Gregory? Are you having any issues? Okay, welcome back, everybody. So let's go through the case study again and talk about some of the answers here. Okay, so this is a story about a woman who went to the eye doctor, which again is something everyone should be doing, not just people who wear glasses or contacts, but everyone should be going to the eye doctor. And that's because you want to make sure you're checking on the health of your eyes. Okay, so the first question, what is central vision, and where is Samantha's central vision focused during that visual field test? So central vision is the area where your eyes are looking straight ahead. And this is the area of greatest visual acuity, or where our vision is the strongest. This is why when you see something moving out of your peripheral vision, your instinct is to turn and look at it directly. And so you're lining up the area of your eye called the fovea centralis, which is the central part of your eye, your central part of your vision. And this is the area that's predominated by cones. So again, C for cone, C for color, C for center. And which part of the retina are the light rays from the object of Samantha's central vision landing? So again, I just said this is the fovea centralis, which is the area that has the highest cone density. What is our peripheral vision? And on which parts of the retina are the light rays from the objects in Samantha's peripheral vision landing? So the peripheral vision are things that are on the side. It's everything when your eyes are looking directly ahead. You can test it by moving your hands slowly back, and you're still aware that something is happening there, but it's not the most sharp vision. Okay, and this is mainly made up of rods. So we have more rods than cones in the center part of your vision, or in the side part of your vision rather, in your peripheral vision. And so they're not quite as strong as that central vision. Okay, so then we go down to question 4. What structure in the eye is responsible for our physiological blind spot, and why does it cause this? This is our optic disc. So your blind spot is called your optic disc, and it's the area where your optic nerve leaves your eye. And so you don't have any photoreceptors there. And this, while it's a physical blind spot, your brain actually fills in the gaps. So you're not aware that you have a blind spot. It's not like when you look straight ahead, you notice like a little black hole of nothing, right? So your brain actually fills in the gaps. And this is something that your brain does all the time with visual information. And so the visual cues are going to go from your photoreceptors in your retina to your bipolar cells, to your ganglion cells. Those ganglion cells will form your optic nerve, and they'll cross back at your optic chiasm. So the information from the left side of your world is going to be processed by the right side of your brain, and vice versa. They'll pass through the thalamus, which is like that relay station, and then they'll go to your visual cortex all the way back in your occipital lobe. And here, your brain makes snap decisions about what you're seeing, and it's going to fill in any gaps. So it's going to take that area where there is a blind spot and fill it in. The reason your brain moves so quickly, or one of the fun things that your brain moves so quickly, that's where you get all of those optical illusions, because your brain is making really fast decisions based on those visual cues. Okay, so I expect that you're able to label the model of your eye. It's very similar to the model that you see in your textbook, so please make sure you're able to do that. And let's talk about our aqueous humor. So aqueous humor is responsible for maintaining our interocular pressure, your eye pressure. So you can think of your eye like a water balloon, and the aqueous humor is the water in the water balloon that gives it its shape. And we have 3 layers of our eye that form the outer casing. And glaucoma is when you get an increase in that anterior chamber. So the aqueous humor is going to be building up. And so what we're doing is we're overfilling that water balloon. And this is usually caused by an issue with drainage. And so normally with your eye, with your aqueous humor, it's going to be in your eye, and it will then drain out and be replaced, and drain out and be replaced. But if the drain is blocked, it's going to build and build and build. And that increase in pressure is a problem because, question 8, what it's going to do is it's eventually going to pinch and damage your optic nerve, and you'll start to lose peripheral vision first, and then eventually central vision. And so you want to catch glaucoma early. And so what they do, and this is breaking down to questions 9 through 11, what they do is they're going to test your interocular pressure with a tautonomy test. Basically, if you've ever been to the eye doctor, you put your face in a thing, and they do a little puff of air, and they measure how that affects your cornea. So again, interocular pressure, question 9, needs to be kept within a certain range. Why? Well, if pressure gets to be too high, it can start to damage the photoreceptors in your retina, and that is bad. So question 10, if the cornea flattens less than normal, so you would expect your water balloon, when you push against it, to flatten a certain amount. If it doesn't flatten enough, that means your interocular pressure is high. It's above normal. You've overfilled your balloon and you can't flatten it. However, if it flattens more than normal, that means you've deflated your water balloon and your interocular pressure is below normal. So both of those tests, both your visual field test and your tautonomy test, provide information about the development of glaucoma. Which of these tests is able to catch glaucoma at the earliest stage? That is the tautonomy test, the air pressure or the eye pressure test. And that's because the first thing that happens is you get this increase in pressure that eventually then starts to damage your photoreceptors. Okay, so what I want you guys to submit, if we go back to the home page here, as I very slowly load. Here we go. So we're all the way here in module 6. What I want you to submit for your class 11 assignment are your answers for part 1 of the case study and the notes you took on those videos that we watched, those 3 videos. Okay. Please be aware that you have your next set of guided reading questions due on Sunday. You have 2 of them, one for vision and one for your other special senses. And next week, what we'll focus on are mainly hearing and balance. But keep in mind, you also need to know information about smell and taste. And then we'll wind down the module, as always, with a homework and a quiz. The last module we're going to get to is the endocrine system, and we're only going to do a 1-week module for this. The endocrine system is something that I feel like is best done kind of at the end of A&P2 because it touches on all of the systems of the body. So we're going to briefly discuss a few key components of the endocrine system, but that will pretty much be it. And then you'll have your final exam, and we'll end in finals week with that final project virtual presentation. Okay, so again, please submit this for your class 11 assignment. And with that, you guys are good to go.